ஓகே, குட் மார்னிங் ஸ்டூடெண்ட்ஸ். இன்றைக்கி நம்ம பார்க்க வேண்டியது என்ன அப்படின்னு பார்த்தீங்கன்னா, ப்ரீ ஃபர்டிலைசேஷன் ஸ்ட்ரக்சர்ஸ் அண்ட் ஈவெண்ட்ஸ் அந்த டாபிக் பார்க்கணும். ஸோ இதில் என்ன சொல்கிறோம் அப்படின்னு பார்த்தீங்கன்னா, கருவுறுதலுக்கு முன்னாடி நடைபெறக்கூடிய மாற்றங்கள் என்னென்ன இருக்கலாம் அப்படிங்கிறது. ஸோ ஹியர் த ஹார்மோனல் அண்ட் ஸ்ட்ரக்சரல் சேஞ்சஸ் இன் பிளான்ட் லீட்ஸ் டு தி டிஃபரன்சியேஷன் and development of floral primordium. It is mostly occur in pre-fertilization. The structures and events involved in pre-fertilizations are given below. So first one is male reproductive part, androecium. Androecium mostly made up of stamens. Here the stamens mostly possess an anther and a filament. The androecium mostly made up of stamens. Stamens, it consists of an anther and a filament. Anther bears pollen grains which represents a male gametophyte. That is, note that point, male gametophyte. In this chapter we have to discuss the structure and development of anther. Okay, the development of anther. First one, the development of anther. A very young anther develops as a homogeneous mass of cells. A homogeneous mass of cells surrounded by an epidermis. During its development, the anther assumes four-lobed structures. It mostly presents four-lobed structure. In each lobe, a narrow or a few rows of hypodermal cells become enlarged with a conspicuous nucleus. It will present. This functions as archisporium. These archisporial cells divide by primordial divisions to form primary parietal cells towards the epidermis and primary sporogenous cells towards the inner side of the anther wall. The primary parietal cells undergo a series of periclinal and anticlinal transverse and vertical divisions and form 2 to 5 layers of anther walls composed of endothelium and middle layers and tapetum from periphery to center, outer layer to center layer. Here, next one is microsporogenesis. Micro means minute, sporogenesis, there is a process of microspore. The stages involved in the formation of haploid microspore from diploid microspore mother cells through meiosis is called microsporogenesis. There is a process mostly occur through meiosis from the diploid microspore mother cells. Okay, this primary sporogenous cells directly are made undergoes a few mitotic division to form sporogenous tissue. The last generation of sporogenous tissue functions as microspore mother cells. Each microspore mother cells divides mitotically to form a tetrad of 4 haploid microspore, that is microspore tetrad condition. In that microspore soon separate from one another and remain free in the another include and develop into pollen grains. The stages in the development of microsporangia is given in figure 1.4. In some plants, all the microspore in a microsporangiums remain held together called pollinium. Pollinium, it will present in calotropis. Here there is a number of microspores, it will present in a pollinium. Example, calotropis. Pollinia are attached to their clamp or clip-like sticky structure called corpusculum. The filamentous or thread-like part arising from each pollinium is called retinaculum. The filamentous or thread-like part mostly arising from each pollinium is called retinaculum. The whole structure looks like inverted letter Y and it is called translator. The Y letter is nothing but a translator. The next one we pass on to T as a mature anther. Transverse section of mature anther reveals that the presence of anther cavity. Transverse section of mature anther reveals that the presence of anther cavity is surrounded by an anther wall. It is bilobed in structure, each lobe having 2 theca, dithecous in nature. A typical anther is tetrasporangiate. The TS of mature anther is given in figure 1.5. First one is anther wall. The mature anther wall consists of the following layers: a epidermis, endothelium, middle layers and tapetum. There are 4 segments or 4 layers mostly present in anther wall. First one, epidermis. It is a single layer and protective in function. The cells undergo repeated anticlinal division to cope up with the rapidly enlarging internal tissues. that is known as the epidermis. There is a single layer. The next one is endothelium. It is generally a single layer of radially elongated cells found below the epidermis. Below the epidermis, there is another layer is present that is called endothelium. The inner tangential wall develops bands. Sometimes radial walls also there of a cellulose are sometimes also slightly lignified. The cells are hygroscopic in nature. Hygroscopic means nothing but நீர் உறிஞ்சக்கூடிய தன்மை. ஹைக்ரோஸ்கோபிக் in nature. In the anther of aquatic plants, saprophytes, clistogamous flowers and extreme parasites, endothelial differentiation is mostly absent in that plants. The cells along the junction of the two sporangia of an anther lobe lack these thickenings. This region is called stomium. Stomium is absent in a mature anther wall. This region, along with the hygroscopic nature of endothelium, helps in the dehiscence of anther at maturity. Okay, the next one is middle layer. That is the third layer is middle layer. 2 or 3 layers of cells next to endothelium constitute a middle layers. These are generally ephemeral. They disintegrate or get crushed during maturity in nature. The cells of tapetum may remain uninucleate or may contain more than one nucleus, or nucleus may become polyploid in condition. It also contributes to the wall materials. Sporopollenin, pollen kit, trypsin and a number of proteins that control incompatibility reaction. Tapetum also control the fertility or sterility of microspores or pollen grains. There is a structure of a mature pollen. There is a mature anther. Here there is a connective. First one connective tissue. Then epidermis is there. Outer layer is epidermis. Endothelium. Middle layer. Then tapetum is there. Stomium. Then pollen grains. Here's a mature anther. There are 2 types of tapetum based on its behavior of this. First one is secretory tapetum, parietal or granular. The tapetum retains the original position and cellular integrity and nourishes the developing microbes. That is called secretory tapetum. Invasive tapetum, periplasmodial. The cells lose their inner tangential and radial walls and the protoplasm of tapetal cells coalesces to form a periplasmodiums. That is called invasive tapetum. The next one is functions. What are the functions mostly present in a tapetum? Here there is a number of functions it will occur. First one is it supplies nutrition to the developing microspores. It will supply the nutrients to the developing microspore and also contributes sporopollenin through Ubi's bodies, thus play an important role in pollen wall formation. The pollen kit material is contributed by tapetal cells and it is later transferred to the pollen surface. Exine proteins responsible for rejection reaction of the stigma are present in the cavities of exine. These proteins are derived from tapetal cells. Then next one is anther cavity. First one is anther wall, then second one is anther cavity. In an anther wall there is 4 layers are there. The next category anther cavity. The anther cavity is filled with microspore in young stages or with pollen grains at maturity. The mitotic division of microspore mother cells give rise to microspore which are haploid in nature. The next one is connective tissue. It is also the column of sterile tissue. It is a column of sterile tissue surrounded by a anther lobe. It possesses vascular tissue. It also contributes to the inner tapetum. It also contributes to the or otherwise functions as a inner tapetum. The next one is microspores and pollen grains. Microspores are immediate product of meiosis of microspore mother cells, whereas the pollen grain is derived from microspore. The microspores have protoplasm surrounded by a wall which is said to be fully developed. The pollen protoplasm consists of a dense cytoplasm and a centrally located nucleus also there. The wall is differentiated into 2 layers, namely inner layer called intine and outer layer is called exine. Here intine is very thin, uniform and made up of pectin. hemicellulose, cellulose, and callus together with proteins. Hexane is thick and is made up of cellulose, sporopollenin, and pollen kit. The hexane is not uniform in nature, it is thin in certain areas. When these thin areas are small and round, it is called jumpos, or when elongated, it is called furrows. It is associated with the germination of pollen grains. The sporopollenin is generally absent in gem spores. The surface of the hexane is either smooth or sculptured in various patterns, rod-like structure, grooved, warty, and punctuate, etc. Like sculpturing pattern is used in the plant identification and classification. Okay, the next one. Shape of pollen grain varies from species to species. It may be globose, ellipsoid, fusiform, lobed, angular, or crescent shaped. The size of the pollen varies from 10 micrometers in Myocytes to 200 micrometers in members of family Cucurbidaceae and Nictagenaceae. Then pollen kit. Next one is pollen kit. The pollen kit is contributed by the tapetum and colored yellow or orange in nature, and it's chiefly made up of carotenoids or otherwise flavonoid. It is an oily layer forming a thick viscous coating over pollen surface. It attracts insects and protects damage from UV radiations also. Okay, the next one is development of male gametophyte. Male gametophyte. The microspore is the first cell of the male gametophyte. It is haploid in nature, and also the development of male gametophyte takes place while they are still in the microsporangium. It will present in a microsporangium. The development it will only occur. The nucleus of the microspores divides mitotically to form a vegetative and generative nucleus. The nucleus of the microspore it will divide mitotically to form a vegetative and generative nucleus. A wall is laid around the generative nucleus, resulting in the formation of two unequal cells. A large irregular nucleus bearing with the abundant food reserve called vegetative cells, and a small generative cell is there. And also there is generally under this two-celled stage, these are liberated. The pollens are liberated from the anther. In some plants, the generative cells again undergoes a division to form two male gametes. The pollen is liberated at the two-cell stage only. 60% of angiosperm pollen is liberated in two-cell stage. Further, the growth of the male gametophyte occurs only if the pollen reaches the right stigma. The pollen, on reaching the stigma, absorbs moisture and swells. The intestine grows as pollen tube through the gem pore. In case of pollen is liberated at the two-cell stage, the generative cell divides into the pollen into the two male cells again, that is sperms, after reaching the stigma or the pollen tube before reaching the embryo sac. The stages in the development of male gametophyte is given in figure 1.6. You can see there is a structure. It will present. In an anther, you can cut the anther. There is a round shaped structure is there. Axon, intestine, and nucleus, gem pore is there. Then vacuole and nucleus. That nucleus is divided mitotically. Dividing nucleus, then vegetative and generative nucleus, irregular. These two nucleus are again the pollen tube is formed generative nucleus and pollen tube nucleus. Then dividing generative nucleus, then tube nucleus also there. Dividing the generative nucleus, again it will produce two cells. Male gametes, tube nucleus also there. Okay, to students, up to this one, today topic is over. Thank you, one and all. Okay.